Hi everyone, so in this video I'll talk about site-directed mutagenesis. So let's get started. So we'll talk about three main topics of site-directed mutagenesis, which is the introduction, the methods of SDM and the need of SDM. So moving with that, so talking about what is site-directed mutagenesis or SDM. So this particular SDM is an artificial in vitro technique for introducing mutation or alteration into the targeted DNA sequence. So site-directed mutagenesis can also be said as a specific location into the genome, and it has been performed by artificial techniques through mutagenesis or introduction of mutations. So site-directed mutagenesis is very simple. So in this particular mutation we have control over the parts of a DNA sequence which we want to mutate or change. So this has a number of advantages over random mutations. So in site-directed mutagenesis, at a specific location on oligonucleotide sequence, the mutation is intentionally created, hence it is also called oligonucleotide specific mutation or site-directed mutagenesis. So we'll talk about all of this in detail. So site-directed mutagenesis has different forms and are carried out in different number of methods, so which we'll be talking about. So we have three different methods for SDM. So one which is the single primer method or oligonucleotide directed mutagenesis, then we have cassette mutagenesis, and then we have the conventional or different sorts of PCR based methods which is carried out during the methods of SDM. So we'll go through all of these three methods for SDM or site-directed mutagenesis. So let's get started with this. So talking about the first method, which is the single primer method. So this particular method is the first method of SDM which was developed, which was the single primer method by Gillam. And in this method, the starting material is a single stranded DNA which needs to be mutated, is cloned into a M13 phage vector. So this is a small diagram that I have adjusted here. I'll show you a bigger picture of this as well. So this is the particular DNA strand, or this is the particular DNA thing that is cloned into a M13 phage vector. So this particular single stranded DNA is the starting material which we want to mutate. So in this site-directed mutagenesis, one thing is very clear that we can insert that particular region that we want to mutate, and that is up to us. So like the same way, we have the single stranded DNA here which is cloned into M13 phage vector here. So as soon as we do that, on mixing this DNA with synthetic nucleotide or primer, the oligonucleotide hybridizes with the complementary sequences. So we definitely use a primer in this case, so which will bind with the DNA sequence. So you can see here, so on the other side, so this was the DNA material with the M13 phage vector, and on the other hand we have the primer here. So this is the chemically synthesized oligonucleotide or a primer that will bind to the DNA sequence. So you can see here, this particular primer binds to the DNA sequence and gets ligated or annealed with the DNA sequence, or binds to the complementary sequence of the DNA. So the synthetic oligonucleotide primes DNA synthesis and itself incorporated into the resulting heteroduplex molecule. So it's a heteroduplex molecule because they are not the same. So you can see here, so these are primed to become, so the primer and the DNA sequence have annealed quite well here to form a heteroduplex. So whenever after we have reached this stage, after transformation of the host E. coli, this particular heteroduplex give rise to homoduplexes whose sequences are either that of the original wild type or containing the mutated bases. So you can see here, it leads to formation of homoduplexes when we transform it in E. coli. So after that we have after that we have reached this stage where it forms a heteroduplex. When we transform it in E. coli, we get homoduplexes in which we get a wild type as well as a mutated form. So in both the cases, both are the same, in which the wild type is the original or the organism's DNA, and the mutated one is the one which we want actually. So the both are structurally the same more or less, That's why they are called homoduplex. And in this case, in the single primer method, we get the mutated form in this two-step simple process. So the frequency with which mutated clone arise compared with wild type clones is pretty low. So the number of mutated clones, so the number of mutated genes we are getting is pretty low as compared to the number of wild type genes. Alright, so the number of wild type clones are more than that of the mutated type. So in order to pick out mutants, the clones can be screened by nucleic acid hybridization with 32 phosphate labeled oligonucleotide as probe. So we can use a method for identification of these mutated clones. So we can use a method of nucleic acid hybridization with using a 32p labeled oligonucleotide as a probe. So this is a very useful method for identifying the mutated clones. So moving with that, so this is a bigger picture of what I wanted to explain. So this is the one that we want. So this is the starting material, which consists of the DNA material, which is cloned into a M13 recombinant phage vector, and this is the primer that we have here, which is annealed here to form something like this. So when it anneals, it forms a homoduplex, which we see here, heteroduplex, pardon. So it leads to formation of a heteroduplex, and when it is transformed in E. coli, which leads to formation of homoduplexes, it forms to leads to formation of a wild type as well as a mutant. So the frequency is less for mutant, whereas the frequency for wild type is more. And in this simple two-step process, we get the mutant genes, which can be screened via nucleic acid hybridization by using a 32 phosphate labeled oligonucleotide as a probe. So this was about the single primer method. So moving with that, so talking about the disadvantages of primer extension method or the single primer method. So the double-stranded heteroduplex molecules that are generated will be contaminated both by single-stranded non-mutated template DNA and that has remained uncopied and by partially double-stranded molecules. So this is a very valid point actually. So whenever the primer ligates with the DNA sequence, so which is the first step when it leads to formation of heteroduplex, so it might be chances, there might be definite chances that it might get contaminated both by any of the single-stranded non-mutated template DNA that has remained uncopied or partially double-stranded molecules. So it can be contaminated by the non-mutated DNA sequences, right? So the ones which are mutated are pretty much safe, but whereas the non-mutated ones can do the damage here. Also, the second point which says that the presence of these species considerably reduces the proportion of mutant progeny. Also, these they can be removed by sucrose gradient centrifugation or by agarose gel electrophoresis, but that is time consuming and inconvenient and costly as well. So the presence of these species considerably reduces the proportion of mutant progeny. So the number of mutant clones that we get via this process is pretty less actually, but the frequency can be increased of course, but we need to use a method known as sucrose gradient centrifugation and agarose gel electrophoresis to check on the gradient again and again and to keep on increasing the frequency. But of course that is time consuming and inconvenient as well as it's bit costly. Also, the third point which says that following transformation and in vivo DNA synthesis, segregation of the two strands of heteroduplex molecule can occur and yielding a mixed population of mutant and non-mutant progeny. Also, the mutant progeny have to be purified away from the parental molecules, and this process is complicated by cells mismatch repair system. So as it says that it's an in vivo DNA synthesis method, so the segregation of two strands of heteroduplex, so when we get a heteroduplex and then we transform it into E. coli, we get a homoduplex in which one of it is the wild type and one of it is the mutated one. So the separation when it occurs, so the separation from heteroduplex to homoduplex, it might yield in a mixed population of mutant as well as non-mutant. So it might be a hybrid of mutant as well as non-mutant. So whenever we get a mutant clone, so there might be chances we might get a half of them might be mutant and half of them might not be mutant. So there is a risk of this particular scenario that might happen. So these are some of the advantages that are shown here for the single primer method. So moving with that, so talking about the cascade mutagenesis. So cascade mutagenesis is very simple, and in particular this particular method in cascade mutagenesis, a synthetic double stranded oligonucleotide is used as a starting material. So in this particular, a small DNA fragment, you can say, is used as a starting material which contains the desired mutant sequence is used. So don't refer to this diagram now, I'll tell you how it is gonna start. So a double stranded DNA is used as a starting material for cascade mutagenesis. So coming to the second point, which is cascade mutagenesis is possible if the fragment of the gene to be mutated lies between the two restriction enzyme cleavage sites. Alright, so this particular point says that we want this particular method of cascade mutagenesis will only happen when we have the mutated gene or the starting material or the DNA fragment which should be present between the two restriction sites, cleavage sites. So let's say this is the two restrictions. So this is a particular plasmid, so this circle is a plasmid, and let's say it has two restriction sites like these. So this, let's say this is eco R1 restriction site and this is HIND3 restriction site. So this particular process will only happen if the desired DNA or the starting material, the small fragment, should be present here. It should be present in between the two restriction endonuclease cleavage sites. So if this particular DNA material is not present between the two restriction endonuclease cleavage sites, this particular cascade mutagenesis will not happen. Alright, so coming to the diagram now. So this particular diagram says that this is the plasmid, as you can see, and this particular plasmid has two restriction endonuclease enzyme cleavage sites. So this is the HIND3 cleavage site and this is the eco R1 cleavage site in a general plasmid DNA. So this is the overall structure we have here. But what we need actually in this cascade mutagenesis, we want that this particular region in between the two restriction endonuclease enzyme must contain the starting material or the DNA which we want to mutate. Alright, so what we can do here is we can remove this particular part here. So this particular part we don't require. So this particular part does not contain the gene that we want to mutate. So what we'll do is we'll initially remove this part. So this would look something like this. So the fragment will be removed and this would look something like this. Alright. And now what we'll do is we'll insert the mutant DNA sequence in between the place that we have removed. So we'll cover that place with the mutant DNA sequence. So we have filled that place with the mutant DNA sequence. So that place is now filled with the mutant DNA sequence which is in between the two restriction enzyme cleavage sites. So as far as we got that, so now that we have the mutant DNA in between the two restriction enzyme cleavage sites, we can carry out this process now. So now that we have got this, we can transform it in E. coli and get the mutant plasmids, which is very simple as the general method of attaining or understanding the mutant plasmids. So we got this mutant plasmid and we are transforming it in E. coli. So E. coli is very versatile, as we know, which is used for checking or determining the mutant clones. So that way we are doing the same here for cascade mutagenesis as well. So moving with that, so talking about the PCR method for SDM, which is the most important method, which consists of three methods, which is the conventional PCR, the nested PCR, and the inverse PCR. So moving with that, so talking about the conventional PCR, so most of us know what conventional PCR is. So the conventional PCR method is one of the most popular and easy methods for site-directed mutagenesis. So in this method, a short single-stranded PCR primers are designed in a manner which contains the mutation. So it consists of primers, forward and reverse primers, which contains the mutations which will cause the amplification and all of that. So also we use a tag DNA polymerase is used in the conventional PCR and does not have the endonuclease activity, hence it cannot identify mismatch during the amplification. So the tagged DNA polymerase does not have the exonuclease activity, so it cannot proofread, so thereby it won't identify the mismatches during the amplifications. And if the mutation is minimal enough to incorporate, the primer beats to the DNA along with mutations. And the newly synthesized strand have the mutation of our interest replaces the original sequences over a period of amplification cycles. So this is a very simple method actually, so in which a piece of DNA is used. So let's say this is a piece of DNA, along with that we use two different types of primers. So let's say this is the forward and this is the reverse primer that is being used. So the primer ligates with the DNA sequence. So this particular primer moves in the forward direction, this particular primer moves in the reverse direction. So it will cover the entire DNA sequence, as you can see. So this is the thing. So this is the, you can say, this is the forward primer, this is the reverse primer, and as the PCR process is being carried out, you can see here the primer contains a mutation. So mutation at the end of the primer. So this particular primer will move or travel throughout the DNA sequence as the PCR product or the PCR cycle is carried out. So this would look something like this, and the mutation will be inserted into the particular region of the DNA in the same way. And also we are using this particular thing, which is tagged DNA polymerase, so that this particular entire process is not proofreaded or the mismatches are not caught. Alright, so the DNA polymerase is used because it does not have the exonuclease activity. So we can use a primer that contains the mutation and we'll get the mutated or the mutations that we want in the particular gene through this process as well, through the process of conventional PCR. So moving with that, so talking about the nested PCR. So nested PCR is a very new type of PCR actually, so which we'll talk about, which is it has two sets of primers are used in a single set of primer is nested. So actually in nested PCR we use two sets of primers. So we use two sets of primers and this particular process is carried out or gets completed in two steps. So both are completed in two steps, and it has also two sets of primers. So I'll show you with the diagram first. So this is the diagram overall that you can see here. So you can see many numberings you can see here that are present here, such as 1, 2, 3, 4. So this particular 1 and 4 are the forward and the reverse primer, and this particular 2 and 3 are the inner forward and the inner reverse primer. Alright, so this particular, so you can just demarcate it like this. So this particular region is the, so you can say this is the first pair of primer and this is the second pair of primer, or you can say first and the number 4 is the first set, or 2 and 3 are the second pair. So you can say whatever you feel like. You just have to remember that there are two sets of primers that are being used in nested PCR. So you can see here, this is the forward primer, this is the reverse primer. Number 2 is inner reverse primer, and number 3 is inner forward primer. All right. So as the PCR is being carried out, what happens in the first step? So in the first step, what happens is this number 2 and number 3 carries the mutation, or this particular primer carries the mutation on mismatch pairs. All right. So these two primers contain the mutations. So what happens in the first step, that these two primers ligate or cut the strands, cut the DNA sequences into two. So this will lead to formation of two different products in the first step. So as you can see, you don't see the number 2 and number 3 here because they have caused the amplification here, which contains, so let's say this particular region contains number 2 and this particular lower part contains number 3. So what happens in the first round of amplification is this particular region of DNA is amplified. So this entire region, so let's say this is the DNA sequence. So this particular DNA is, when you amplify it, when you break it into two, you get two pieces of DNA, right? So you get like this, you would get something like this, you get some two pieces. This is the first piece and this is the second piece. So the same thing is happening in this process as well. So you can see you are getting two pieces of DNA here, cause the ligation has happened. Why? So the question is why the ligation has happened. So the answer is because the number 2 and number 3 carries the mutation. Alright, whereas number 1 and number 4 do not carry any mutations. They are just for annealing with the DNA sequences, right? So number 2 and number 3 carrying the mutation, they have amplified that particular region that are common to both. So it leads to formation of two different products in one. And in the second round of amplification, what happens is number 1 and number 4 primers, so the forward and the reverse primers, ligate the two regions together. So the mutations which are formed here, so the mutations that are inserted into the DNA via number 2 and number 3 primers. So these two primers have inserted the mutation. So these two primers have inserted the mutations in the DNA sequence. Alright, so these two primers have inserted the mutations in the DNA sequence by amplifying it. Now number 1 and number 4, so the forward and the reverse primer now ligate these two sequences so that the mutations are inserted overall and they are formed as one. So you can see here the mutations are inserted here as well as here. So I'll use an eraser for your purpose, so that everything becomes clean. So yeah, so you can see here, so I use a black pen so you can see. So this was the initial thing. This is the second step. All right, so in this what happens is this, so when the amplification has happened by number 2 and number 3 primers, this has inserted the mutations by amplifying it. So when they amplify it, they insert the mutations in that particular DNA strand. Alright, so this is the mutation that has been added. This is the mutation that has been added. Alright, so these two mutations are added by number 2 and number 3 primers. So these needs to be ligated so that we get a ligated form, mutated form of the DNA. So the ligation is happened by number 1 and number 4 primers, or the forward and the reverse primers. So this is the entire concept of nested PCR. So that is only written here. All right, so you can just give a read here. All right, so moving with that, so talking about the general outline of SDM. So the general flow of experiment is as follows. So the designing the primers for introduction of specific mutations and checking all the primer parameters. Then we have synthesis of the oligonucleotide primer artificially, then preparing PCR reaction based on the methods listed above, performing the PCR reaction, authenticating the amplicons using the agarose gel electrophoresis. We have purification of amplifon by DNA purification kit. Then we have DNA sequencing for verifying the mutagenesis result, and then they send the sample for cloning. So that's a hell lot of process that is being carried out in a SDM. So you can see here there are different methods that are being carried out using SDM. So first of all, we went through single primer method, then we have the cascade mutagenesis method, then we have the PCR method. So talking about the need of single site-directed mutagenesis. So the site-directed mutagenesis may use to generate mutations that may produce rationally designed protein that has improved and special properties. Also, specific mutation specific mutation in DNA allows the function and properties of a DNA sequence or protein to be investigated in a rational approach. So the tool used in the study of gene characteristics, in which by introducing gene specific mutations, the properties of DNA and all of that can be studied, and proteins may be engineered to produce proteins that are tailored for specific amplifications, and it is also used in the screening of SNPs. So let's just keep this video till here. Hope you enjoyed this video, and thanks for watching.